So thanks to the organizers for the opportunity to share my work in this amazing symposium. I would like to share today how by transferring cerebrospinal fluid from young to aged mice, I became very interested in oligodendrocytes in the aging brain. I'll just start by mentioning that aging is the primary risk factor for major human pathologies, including many that we've heard today about, like cancer, diabetes, cardiovascular disorders, and neurodegenerative diseases. And this notion has led to extensive research to develop strategies in model animals to control the rate of aging, like we've just heard from Raz, and in some cases to seemingly reverse many aging hallmarks. One such rejuvenation strategy is achieved by the surgical connection of a young and aged mouse in a process termed heterochronic parabiosis, or by performing repeated plasma transfers from young to aged mice. These studies done by the Weiss-Curie lab and others have demonstrated that these blood-borne proteins hold the potential to restore various aging hallmarks in multiple tissues, including in the brain. However, the brain is unique because it's partially secluded from the plasma by the brain barriers, which makes it very challenging to identify what are the ways by which these useful proteins interact with the cells in the brain. Instead, the brain is bathed in cerebrospinal fluid, or CSF, which flows within the brain ventricles and surrounds the brain and the spinal cord. And the CSF is composed of a unique set of ions and proteins that match the brain's unique needs. So when I started my postdoc, I wondered, could CSF be used to rejuvenate aged mice? And if so, would it simplify our understanding of how these proteins interact with the cells in the brain? So to do it in a functional way, I collected CSF from young mice, directly from the brains of young mice, and infused it to the lateral ventricles of aged mice using mini-osmotic pumps. These CSF transfers haven't been done mostly because of technical difficulties in the collecting enough CSF without blood contamination. We then screened these aged mice at various levels to identify what are the cells or cellular pathways that are mostly affected by these manipulations. At the behavioral level, we assessed a long-term memory recall in a paradigm of remote preconditioning recall. Mice are put in a chamber where they receive a food shock in a certain context paired with a flashing light and a tone. They're then infused with young CSF or artificial CSF as control. And 3 weeks later, they're put in the same context where their recall is assessed by their levels of freezing. And indeed, mice that receive young CSF retain the memory better than controls. Because this is an hippocampal dependent memory task, we isolated the hippocampi and performed RNA sequencing. And to our surprise, many of the differentially expressed genes belong not to neurons or microglia, which are cells that are studied mostly in the aging brain, but rather to oligodendrocytes. And these genes were mostly upregulated, as you can see in the violin plot, and consisted of major regulators of oligodendrocyte maturation, as well as myelin components such as MBP and MAG. And this was intriguing to us because MRI studies of healthy humans throughout their lifespan show that white matter volume increases in the first 5 decades of life, so well beyond the development of the brain, but then unfortunately declined dramatically in a process that seems to be very correlated with loss of cognitive function. And this white matter is formed by mature oligodendrocytes, which wrap their membranes around axons to support neuronal functions in various ways. They are produced in a process termed oligodendrogenesis, where oligodendrocyte progenitor cells, or OPCs, given the right cues, separate and mature to form this myelinating oligodendrocyte. And interestingly, this process of oligodendrogenesis was recently shown to be necessary for formation of long-term memory consolidation and recall in young mice. Yet we don't know much about how these cells age and whether they are causally linked to aging-related cognitive decline. So we stained the young and aged mouse hippocampus with an OPC marker, seen here as green dots. And as you can see, while the cells remain in vast numbers in the aged brain, when we pulsed them with a marker for proliferation, we saw that their proliferation rate declines dramatically with age. So we hypothesized that there might be a factor in young CSF that is boosting their proliferation and maturation. So to check that directly, we infused mice with young CSF and pulsed them with EDU or BRDU to label proliferating cells, and indeed found that that boosted OPC proliferation in the aged hippocampus. In another cohort, we let these mice, we let these cells to mature for 3 weeks, and indeed found an increase in myelin in the molecular layer of the hippocampus. So how does young CSF promote myelination? We turned to a cell culture system in a collaboration with the Zucker lab at Stanford and added CSF and performed bulk RNA sequencing after an hour and 6 hours to see what are the very initial cellular responses to young CSF in OPCs. And indeed, the top differentiated gene was an immediate early gene and a transcription factor called serum response factor, or SRF. And SRF is known to integrate various extracellular signals to drive expression of cytoskeletal genes, which in turn mediate cell migration, proliferation, and differentiation. And indeed, at the 6-hour time point, while SRF was back to baseline, many of its known differentially expressed or many of its known gene targets were upregulated. And this transcription factor was not studied in oligodendrocytes before, and we're currently following up on that on a different study to identify what are the roles of SRF in oligodendrocytes. Back to the aging brain, we stained we measured the levels of SRF in hippocampal OPCs, and indeed found that they were downregulated with age. So this really set us to find what is the factor in CSF that is inducing this effect, to try to restore SRF levels and oligodendrocyte function. So we turned back to an in vitro system where we used SRF activation as a readout to screen for for CSF proteins that are known to be present in the CSF and find the ones that could activate SRF. And indeed, one of the top candidates was a fibroblast growth factor called FGF17. FGF17 is known to be a brain-enriched protein, and it's expressed very early on in brain development, where it coordinates the formation of hindbrain structures, yet not much is known about whether it's even expressed in the adult brain and by which cells and what are its functions. So we performed RNA scope and stainings for FGF17, and to our surprise, found it very abundantly in cortical and hippocampal neurons in young mice, yet its levels dropped to almost 0 in the aged mouse. We next mined human CSF proteomic datasets and found that FGF17 levels decreased with the age in the healthy population. So this really begged the question, would FGF17 be sufficient to mimic the effects of young CSF? So we infused the recombinant mouse FGF17 to aged mice, very similarly to the young CSF studies, and found that it's sufficient to promote OPC proliferation in the hippocampus. And most importantly, it mimicked the effect that we found with young CSF on long-term memory in the exact same paradigm. So overall, these results inform the following model, where oligodendrogenesis declines with age, and re-exposure of the brain to young CSF or to FGF17 is sufficient to promote oligodendrogenesis. And this is concomitant with improvements in long-term memory recall. And I think that overall, this study supports the idea that CSF has a functional impact on brain function, and its changing composition with aging might result in cognitive decline. It also highlights the importance to study oligodendrocytes in aging and neurodegenerative diseases. And I find it very encouraging that exposing oligodendrocytes to young CSF proteins, even at very late stages of aging, is sufficient to partially restore their functions. And I'm very excited about these directions to follow up in my future lab. And so with that, I would like to thank Tony for being an amazing mentor, and all the lab members contributed to this work, especially my trainees throughout the years, and to all of our collaborators and funding sources.